Chapter 19 in your book is titled Patterns of Chromosome Inheritance, and in this chapter we're going to be looking at the structure of DNA and the processes of cell division, both mitosis and meiosis. And we're going to be doing that looking at sections 1 through 6. And in order to make sure that I get through everything I need to, I'm not going to read our takeaway questions out. If you want to pause the slide here and jot those down, feel free to do so. As we recall from our discussion of the reproductive system, the only cells in our bodies that are haploid, having only a single set of chromosomes, are the gametes produced by meiosis, sperm cells in males and egg cells in females. The rest of the cells in our bodies, the ones carrying out all of the other body functions that we've discussed this semester, are diploid, meaning they contain 2 full sets of chromosomes. And these cells, these diploid cells, duplicate by mitosis to produce new cells so that organisms can grow, replace damaged cells, and also repair damaged or worn out tissues. So you can imagine that's probably happening in us all the time because those are things that we're experiencing regularly. If 2 haploid gametes that we saw formed by meiosis come together by fertilization, they then form a diploid zygote. And these diploid cells are also called somatic cells. And somatic cells have 2 sets of chromosomes. So they have a complete set of 46 chromosomes in 23 pairs. We'll look at why that's so important when we get to genetics. Cell division by mitosis is what allows a newly fertilized zygote to add new cells and grow throughout the process of fetal development. And then an infant continues to grow because its cells are continually undergoing mitotic cell division. And then after you reach your adult size and stage of development, your cells continue to divide by mitosis in order to repair damaged cells. So mitosis is really happening in each of us right now. The chromosomes that we were talking about are housed within the nucleus and include DNA and a protein called histones. And histones are the proteins that allow DNA to wind up on itself, whether it's going to be in the chromatin state or the chromatin state or the chromosome state. And we'll see why that's so important when we get to the process of mitosis and meiosis. And then when we refer to a gene, we mean a segment of DNA, a code that codes for a specific trait. And we're going to look at genes in a lot more detail in that specific code when we get to that process of transcription and translation, where our DNA is actually replicating itself. So when we talk about chromatin, we mean uncondensed DNA, not wound up around that histone proteins. So when DNA is wound up, that is condensed DNA. When it is unwound, free of those histones, we're going to call that chromatin, uncondensed DNA. Chromosome is when it is condensed and wound around those histones. And then we have another vocab term here, chromatids. A lot of these are very similar, sister chromatids, and that's referring to exact copies of DNA after the DNA has been replicated. So you're going to have a chromosome containing DNA, but at some point during the cell cycle, which we'll look at here next, you're going to need to replicate that DNA so that you have enough to pass on to that new cell that you're creating by mitosis. So that replicated DNA are called sister chromatids, exact copies of one another. And the chromatids are going to be connected together by the centromere, the thing that connects the 2 sister chromatids together is the centromere. And then when it's condensed down, we know that that is called the chromosome. And when that cell does finally undergo replication, that will be in the DNA will be in the chromosome state, wound around those histone proteins. Humans have 23 homologous pairs of chromosomes. We have 22 autosome pairs. So those are going to be the things that code for the traits of our physical selves, and then the things that code for the proteins that are fueling all of the physiological processes that we've talked about in all of those other body systems. And then we have a single pair of sex chromosomes that allow us to produce the individual sex hormones and lead to our external sex characteristics, whether that be male or female. This image that you see on the slide is called a karyotype, and it's an image, a stained image of all of the chromosomes from a single individual from a single cell. And the karyotype on this slide, we know this, if you were looking at these two right here, that would be indicating a male individual, having one X chromosome and one Y chromosome, receiving an X chromosome from their mother and a Y chromosome from their father. So the sex chromosomes in a sperm cell that fertilize the egg is what will determine the genetic sex of the offspring. If the karyotype ends up looking like the one inside the box here, 2 X chromosomes, then that will lead to a female individual having one X chromosome from mom and then getting that other X chromosome from dad. Each one of our cells undergoes what we call the cell cycle. It's going to spend the majority of its time in what we call interphase, and that means that it's not undergoing cell division. During interphase, our DNA is primarily in the uncondensed state, so not wound around those histone proteins and available for transcription and translation, so that that DNA code can actually be used to fuel our body's processes. Interphase can be broken down into 3 kind of subphases, kind of preparing the cell for when it undergoes mitosis. So we first have the G1 phase, and during G1, the cell physically grows larger, so you get more cytoplasm, and then all of the organelles that we talked about, the endoplasmic reticulum, the Golgi body, all of those are duplicated. During the S phase of interphase, DNA is replicated inside the nucleus. So that's where we're going to go from our single chromatid to replicating it into 2 sister chromatids connected by that centromere. That has to happen before we even enter the process of cell division, so that the cell has enough DNA to pass on and have to create 2 exact copies of itself. And then finally, the third phase of interphase is going to be the G2 phase, where the cell is going to continue to grow and become physically larger, adding more cytoplasm, so that when the cell does undergo that process of mitosis, there's enough stuff there to physically create 2 new cells. Mitosis itself is that process of cell division. So if you're looking at the cell cycle, you can think of it as being in 2 phases. Interphase, where all of those things are happening, and then cell division. And cell division is then going to be broken down into 4 of its own phases. But the goal of mitosis is to take a single nucleus and divide it into 2 identical nuclei. So you're going to start with a single nucleus containing 2 copies of all of the genetic information. And the end result is going to be 2 independent nuclei, each containing a full set of DNA. And we will cover the phases of mitosis in the separate video so that they're not split up. See you there.